Welcome to today's HealthLine webinar from the American Association of Kidney Patients, titled Advancements in Kidney Transplant Research and Innovation. Thank you for joining us for this special webinar as we close out National Donate Life Month. My name is Jerome Bailey. I am AAKP's Director of Patient Engagement and Advocacy, and I am pleased to be the moderator of today's program. As the oldest and largest fully independent patient-led kidney patient organization in the U.S., AAKP is proud to host our webinar series as a service to our fellow kidney patients, family members, and care partners, healthcare professionals, researchers, and policymakers. AAKP's HealthLine and HealthLine Innovator webinars fall under our Center for Patient Research and Education. We believe patient and care partner education is an integral part of an individual's treatment, and to that end, AAKP is a champion for full patient care choice, the protection of the patient-doctor relationship, and the elimination of artificial barriers and government determinants of health that impede patient care choice and access to available treatment options. At AAKP, we work to ensure that patients have a central role in the research and innovation process so that unmet needs are identified and unique patient insights and lived experiences fully inform the research and the product development lifecycle. This includes clinical trials to regulatory action and payment decisions. The field of kidney transplant innovations has seen significant advancements in recent years, offering hope for the development of novel treatments for kidney disease that can address the pressing needs of our community. Today's exciting topic has gained momentum and is the focus of much interest for AAKP. As researchers continue to push the boundaries of medical science, the field of kidney transplant innovations is poised for even greater advancements in the future. With a growing emphasis on personalized medicine and cutting-edge technologies, the possibilities for improving outcomes for patients with kidney disease are endless. We are thrilled to present today's program featuring top allied experts in the field of kidney transplantation who are committed to developing solutions to the global transplant shortage while transforming the treatment of kidney failure for patients and their families alike. The presentations and discussions you will hear will shed light on challenges and opportunities and alternatives to human organs for transplantation, as well as potential impact of innovative treatments on patient outcomes. We are confident that this webinar will inspire new ideas and collaborations to drive progress in the field of kidney treatment options. Leading up to this webinar, AAKP conducted an innovation flash survey through our Center for Patient Research and Education. I am delighted to share some of the results with you that will help set the stage for our esteemed speakers. As you can see, this survey reached across our network to engage a highly diverse group of respondents. This slide is significant for more of what it doesn't say than for what it does. As you can see, many of the respondents are knowledgeable about alternatives to dialysis. However, what you can infer is that the kidney community, led by patients and their family members, are equally invested in finding new treatment solutions. These percentages show that patients are monitoring what research is being done. This slide depicts what research underway is highly supported by the patient community. At 74% and 55% respectively, patients are cheering on efforts that support bringing an artificial implantable kidney and xenotransplantation to fruition in the near term. Our next slide took a deep dive into xenotransplantation, asking if you consider a transplant if it was a safe, FDA-approved treatment. An overwhelming amount at 74% said yes. And then we asked, what is most important for you in considering accepting a xenotransplant? The top three being positive clinical trials, knowing that there is a potential that xenotransplants may not require lifetime immunosuppressant medication, and support and approval from the FDA. And finally, we asked a very simple question. Do you believe that research on xenotransplantation should be accelerated so that more choices are available for those on dialysis? We nearly broke the response rate with an overwhelming 95% saying yes. This is all very exciting and important insights into the commitment patients are willing to make to support alternatives to dialysis. Patients want more, they want better, and they deserve it. Our first speaker is Dr. Mike Curtis, President and CEO of eGenesis. With over 30 years of experience in scientific research and leadership and biopharmaceutical drug development across various therapeutic areas, Dr. Curtis is at the forefront of innovation. At eGenesis, he spearheads the company's mission to address the global transplant shortage and revolutionize the treatment of organ failure. Dr. Curtis, thanks for joining us. It's my pleasure to turn the presentation over to you.
So thanks a lot for the invitation to join you today and the interest in what we're doing here at eGenesis. We're thrilled to be able to bring our technology to patients who are in tremendous need, are suffering from solid organ failure. We've made tremendous progress over the past 5 or 6 years, and we're thrilled to be now bringing our products for the first time into patients. So at eGenesis, we've developed a novel platform that includes porcine genome engineering to produce organs that are more compatible with human transplant recipients. And the goal here is really to end waitlist mortality and then to bring the benefit of transplant to a much broader group of patients. As we know today, we're constrained with organ supply, which creates a transplant waiting list. With an endless supply and renewable supply of compatible organs, we can actually bring transplant to a much larger group of patients. And in doing so, create new modalities of treatment for everyone suffering from end-stage organ failure. So we're focused on really 3 major transplant areas: kidney transplant, heart transplant, and what we think of as liver perfusion. And over the past 7 years, we've made significant progress across all 3. And I'll mention the liver and heart program first. I know we're here primarily to talk about kidney, but we've made progress in liver perfusion. This past December, we did the first ever transgenic porcine liver perfusion study in a patient that was in brain death at the University of Pennsylvania, achieving over 3 days of continuous perfusion. And the idea here is to use this technology to support patients suffering from liver failure. And then we have a collaboration with David and John Cleveland, 2 pediatric heart transplant surgeons, where we've transplanted our transgenic hearts into baboons, achieving over 250 days of post-transplant survival. The idea here is to use the porcine heart in the pediatric setting as a bridge to human transplant. So children that are suffering from end-stage heart failure will often pass away prior to having a chance to receive a human heart. The idea here is to use a porcine heart to bridge them. And then most recently on our kidney program, we completed the first ever porcine kidney transplant in a living person. This was in collaboration with the transplant team at Mass General. And the patient was Mr. Rick Slayman, a true pioneer in the field of kidney transplant, agreeing to be the first patient to receive this therapy. And I'm happy to report today, Mr. Slayman is doing well. He's about 37 days post-transplant. The kidney's functioning well, and he's recovering. So we're thrilled with that first step. And really what's enabled all of these advances is the production of this animal, right? So this is the most highly engineered animal in history. It carries a total of 69 edits to the porcine genome. And when we approach engineering the porcine genome, we really have 3 things in mind. One is to reduce the risk of pathogen transmission from the donor to recipients. In part to do that, we engineer or inactivate endogenous retroviruses that are present in every genome. We use technology called CRISPR-Cas9 to inactivate all 59 copies of the virus in the porcine genome. We inactivate enzymes responsible for the carbohydrate differences or sugar differences between pigs and humans that leads to hyperacute rejection. And so by making these modifications, we eliminate hyperacute rejection. And then we add 7 human regulatory transgenes that promote long-term graft function and improve compatibility of the porcine organ with the human recipient. I think this so far has been bearing out positively in Mr. Slayman's transplant. So the animal carries a total of 69 edits. We produce it in a clean environment. We test it rigorously for pathogens to ensure that we're not transmitting pathogens from the porcine donor to recipients. And this has really enabled us to advance all three programs. This past fall, we published in Nature a series of primate studies. So kind of what led us to actually being able to do the transplant with Mr. Slayman. It was this work here where we studied the porcine kidney in monkeys. This summarizes a total of about 25 transplants. 15 of those were done with the same porcine donor that was used in Mr. Slayman's transplant. As you can see from the blue line, we were able to achieve over 2 years of post-transplant survival in the monkey, which to us gave us the confidence to move forward with clinical studies. The field of xenotransplantation over the past few years has been starting to move these porcine organs into patients. Many of the studies have been conducted in patients that we refer to as decedents. So these are patients who are in brain death. They may otherwise be an organ donor, but for some reason are disqualified from organ donation, so their families consent to whole body donation, and that allows us the opportunity to study these organs in these patients. Just this past fall, Bob Montgomery and the team at NYU performed a 61-day kidney transplant in a decedent. Work has also been done in living patients. Back in 2022 and 2023, Mohammed and the team at Maryland did 2 porcine heart transplants in 2 living patients. We refer to these as the Fawcett and Bennett transplant. Right? And this was showing that we could, working with the FDA, do transplants in these patients under what we call expanded access. So this is when we identify a patient who's in dire need of help. They've really exhausted all treatment options. And this gives us the opportunity and the patient the opportunity to try one of these experimental therapies. So the work that was done at NYU and Maryland and by other groups really led the way for the transplant that we did at Mass General in March with Mr. Slayman, which we did under expanded access, really leveraging all the advances over the past couple of years. And hopefully, maybe many of you have seen this. Mr. Slayman the transplant was done on March 16th. Mr. Slayman was discharged from Mass General on April 3rd. He continues to do well, and we're really thrilled with the outcome. And again, we have a tremendous gratitude to Mr. Slayman for agreeing to participate in the study, as well as the team at Mass General, for which we've had a long collaboration over the past 5 or 6 years. So we're, again, thrilled with the progress, and our intention for the rest of this year is to continue to explore. Unfortunately, there's a lot of patients like Mr. Slayman who have exhausted treatment options or losing access to dialysis and really need a transplant. And because of the current constraint on supply and the way the current allocation is done, those patients are very unlikely to have the opportunity to get a cadaveric kidney. And unless they have a living donor, they really run out of options. So here, with the first porcine kidney transplant, it really gives now a new option for patients in that situation. I'd like to thank everyone again for taking time to listen to the eGenesis story. We're thrilled about the progress that we've made over the past few years and look forward to the 2 or 3 years ahead of us, where we expect to bring our kidney, liver, and heart programs into clinical studies.
Thank you for a great presentation. I know we are all excited with the progress eGenesis is making related to xenotransplantation. I do have a few questions that I'd like to pose to you. AAKP is a major advocate for artificial kidneys, including xenotransplantation. Our strategy is all of the above when it comes to new innovations that can provide life-saving care for those suffering from kidney failure. Generally speaking, as a leader in the field of xenotransplant, are you more optimistic today about the future of kidney innovation than you were a decade ago?
Oh, yeah. Thanks for the question, Jerome. Absolutely. I mean, I think all the advances that we've made in cross-species transplant, even most recently with the transplant with Mr. Slayman, I think is just the beginning of the advances we're going to continue to see in cross-species and xenotransplantation. But I think importantly, those technologies will continue to evolve. Our ultimate goal here is, in the case of xeno, is to produce organs that don't require immunosuppression. I do think there's great promise for some of this, stem cell derived therapies, some of the artificial systems. I think there's great potential there. And I think one of the things we've clearly learned in the field of xenotransplantation is you never say never. Some of these things take longer than you wish they would, but I think there's been really breathtaking advances. And I think that trend is just going to continue.
On March 16th, the National Kidney Month, for the first time, America learned a new kidney from a gene-edited pig was successfully transplanted into an active patient. As you mentioned, the pig from which the kidney originated was from eGenesis. Can you share some background on this story and what it means for the future of xenotransplantation?
Yeah, so when we entered the field of cross species or xenotransplantation several years ago, we were in a unique position to be able to integrate many of the advances that were discovered over the past 30, 40 years. And so the donor animal that we used in Mr. Slayman's transplant really incorporated all those learnings, right? So the donor carried a total of 69 edits to the porcine genome. 59 of those were to inactivate endogenous retroviruses that pose a risk of zoonotic transmission or a pathogen risk to patients. We then also knock out antigens that can lead to hyperacute rejection. We call that the triple knockout. And then we add 7 regulatory human transgenes to control things like coagulation, complement activation, and then regulating mechanisms of innate and adaptive immunity. So it's if it is the most, as far as we know, highly edited mammal in the world, and we think that's really important. And we've shown this in monkeys to leading to long-term transplant outcomes. And when we think about kidney transplant, we want these kidneys to function for years, and we showed in monkeys that we got over 2 years of post-transplant function, and we're hopeful that we'll see the same thing in the first patients.
What type of patient would be qualified to receive a xenotransplant, and how long would a transplant last?
Sure. We're positioning this at this stage is to offer this to patients who have really run out of treatment options. And if you look at actually Mr. Slayman's story, he had a previous allo transplant for about 5 years. And then when that graft failed, he found himself back on dialysis. And he was beginning to lose access to dialysis. So he was running out of the ability to be dialyzed. And then he didn't have the way the current cadaveric kidney allocation works is he would be a very low priority to get a cadaveric kidney. So his treatment options are running out. And if I think you look at patients like him, across the spectrum, there are many folks who will find themselves running out of access to dialysis and don't have access to a transplant. And so we view this population as the right population to bring this technology to first. And I think initially we're looking for and hopeful that we'll get at least 1 year of post-transplant graft function. Again, if the non-clinical studies or the monkey models are a predictor, we're hoping for many years of post-transplant graft function from this donor. And then I think as we demonstrate safety and function in this patient population, then we would be moving into earlier lines of therapy. So patients who may be newly diagnosed, or maybe patients who haven't failed the previous allo, but are getting their first transplant. And ultimately, we'd like to see xenotransplantation move into what we call the front line. So patients who do have end-stage renal disease may be able to get a porcine kidney earlier than they could in the current allocation system.
Lastly, and most importantly, what role do you see for advocacy organizations like AAKP when it comes to getting xenotransplant into the marketplace? Do you envision advocacy organizations being partners throughout the clinical trials, regulatory reviews, and payment decisions before the Center for Medicare and Medicaid Services and insurance companies?
I think it's all the above. I mean, when we come to work here at eGenesis, our focus is on building technology for patients. And I think the patient voice in all of what we do is incredibly important. I actually think the progress that we've made so far and the ability to offer Mr. Slayman this option of a porcine kidney is in part driven by the work of advocacy. Right? So we've been speaking to patients for years about their willingness to participate in these types of studies, what they would need to see to feel confident. And the technology is a little complicated, right? And so I think we view advocacy as a really important partner in helping to explain to the larger patient community what we're doing. And then I also think it's really important to convey to the regulators, the payers, of just what the patient journey actually looks like. Right? Because if you haven't, been a patient with end-stage renal disease, if you haven't spent any time on dialysis or haven't been a transplant recipient, you don't really know what it is, right? And so I really think it's important that the patients are able to tell their story and have an input on all of the players in the space. And so we definitely look to the advocacy to help us bring these products to patients.
This has been a great discussion, Dr. Curtis. Thank you for joining us and sharing the work of eGenesis. Our final presenter is a good friend and ally to AAKP and a two-time KidneyX prize winner, Dr. Shuvo Roy. Dr. Roy is a professor at the University of California, San Francisco Department of Bioengineering and Therapeutic Sciences. He is also a founding member of the UCSF Pediatric Device Consortium, which aims to expedite the development of cutting-edge devices for children's health. One of Dr. Roy's most significant roles is serving as the technical director of the Kidney Project, a groundbreaking initiative focused on developing a compact, surgically implanted, freestanding bioartificial kidney to treat kidney failure. He, along with another good friend and ally of AAKP, Dr. William Fissell, medical director out of Vanderbilt University, lead a team that is dedicated to making this technology a reality for kidney patients. AAKP announced its formal partnership and support of the Kidney Project in 2018 and are committed to ensuring the success of this novel innovation. Dr. Roy, we welcome you. I turn the presentation over to you.
Many thanks to you and to AAKP for hosting this event. My team and I from the Kidney Project are pleased to provide an update on our work towards building an implantable artificial kidney. So today I'll talk about the progress we've made over the years that includes a team from UC San Francisco, Vanderbilt, patient groups like the AAKP, and other researchers both across the country and some from across the world who have been helping us over the last 10 years or so. Our vision is for a universal donor kidney that overcomes the scarcity barrier of transplant while providing its benefits. This device, an implantable bioartificial kidney, is powered by blood pressure alone, no batteries or external connections needed. The device is a two-stage system that replicates the main functions of a kidney. The first stage, the hemofilter, is built from highly efficient semiconductor silicon membranes, which filter toxins from blood using the patient's blood pressure alone, and without the need for blood thinners. The other main functions of the kidney is replicated by the second stage, a bioreactor that also uses silicon membranes to encapsulate growing renal tubule cells while protecting them from the patient's immune system. The bioartificial kidney is a compact device connected to blood vessels and to the bladder using a surgical procedure similar to kidney transplant. To miniaturize the bioartificial kidney, we use silicon semiconductor technology. As we can see from the development of cell phones and other microelectronics, silicon technology allows for extreme miniaturization of sophisticated systems while leveraging almost 75 years of reliable and cost-effective manufacturing. With silicon production techniques, we can now create nanoscale features comparable to sizes of biological components inside cells. Today, I'll provide an update from experiments that we conducted to advance the bioreactor component of the bioartificial kidney. This is the unit that houses the renal tubule cells. We wanted to test whether the features of the precisely engineered membranes will allow the bioreactor to provide immunoprotection, meaning no need for anti-rejection drugs, and prevent the passage of cells and viruses between the patient and the device and the components inside it. In this experiment, we used human kidney cells inside our device prototype. As you can see, it was implanted into pigs, and to test our hypothesis, no immunosuppression was administered to the animals during or after surgery. Now, here are the results from our experiment. The images on the left and the graph in the middle show that the kidney cells inside the bioreactor were alive despite no immune suppression. On the right, the graph shows that the cells express the genetic markers of human kidney cells without any evidence of damage after implantation. This exciting data allows us to envision that the bio-artificial kidney, once it's fully developed, should not require the patients to be treated with anti-rejection drugs. Next, we'll continue to build on these results and advance the bioreactor work by testing the prototypes for longer periods of time and evaluating the actual functionality of these cells. Thank you for allowing me to provide this update for the community. Let me thank those who have contributed to our work, both financially and otherwise. My team at the Kidney Project and I are exceedingly grateful for that support. We also appreciate our partnership with AAKP and the support for creating artificial kidneys within this decade of the kidney. Thank you.
Thank you, Dr. Roy, for another great presentation that provides us with an update on the progress of the Kidney Project. As with other speakers, we have a few questions that we'd like to ask you. How does an artificial implantable kidney function compared to a natural kidney?
Thank you for that question. We designed the artificial kidney to replicate the native kidney. So it's got a filtration unit very much like our own native kidneys, the function that dialysis tries to replicate, but it's coupled with another section that contains kidney cells, something that dialysis does not. It replicates the functions of the kidney tubule. And these two units work in tandem to provide total renal replacement such that the patient is not needed to connect to an external unit, can eat or drink freely, and overall gets treatment 24/7.
When do you think this technology will be ready for human trials?
That's an excellent question. We are doing the research and development to prove that our concept is feasible. To move this to human studies, we'll have to work very closely with manufacturing partners, as well as the FDA, to gather the data, build prototypes that we can then do control testing that's needed before the FDA will let us human studies. We're hopeful that by the end of the decade, given that this is the decade of the kidney, we'll be able to get to the patient community.
Will I need to take immunosuppressant drugs after receiving an implantable kidney?
Sure, immune suppression drugs are critical whenever one gets a transplant organ. However, the immune suppression drugs have a number of adverse side effects. When we began to work on the design of the bioartificial kidney, one of our design criteria was how can we eliminate the need for immune suppression drugs or certainly minimize the amount that will be needed. The membranes we've developed serve as a barrier for the immune system of the patient to interact with the cells that are inside our device. This physical barrier prevents immune rejection. And what our experiment showed was by putting human kidney cells in a pig with the membrane barrier, we saw no evidence of rejection, a very promising sign that in the future we can avoid the need for any immune suppression even after implantation.
And finally, what do you expect the lifespan of an implantable kidney to be? And are there any considerations for potential replacements in the future?
Like any medical device, there's always the chance that there may be the need for a revision or parts may not last forever. However, by working with the right engineering team, material scientists, surgeons, and nephrologists, we hope to make a permanent device, similar to what pacemakers are today, similar to what heart valves are today. Being engineers and scientists, we can prepare that there still may be a failure, and in that case, we hope we'll have developed techniques to minimally invasively replace the components in our device or replace the whole device altogether.
Dr. Roy, thank you for joining us and providing the latest on the Kidney Project and outlining the necessary steps to bring this innovative solution to patients in need. Your presentation was highly informative and greatly appreciated as always. If anyone watching has additional questions for our speakers or about today's webinar that we were unable to get to, please email us at info@aakp.org, noting the webinar title in the email subject line. In closing, if you are not already a member of AAKP, we encourage you to join. AAKP offers free memberships to patients and their family members, as well as living kidney donors. To become a free member, join online at aakp.org/join or by phone at 1-800-749-2257. To receive all the benefits of membership, please be sure to include your email address when signing up. As an AAKP member, you will be notified about all of AAKP's upcoming events and the latest in our educational program series. And when opportunities arise where your opinions and experiences are needed to help inform innovation, advance care, and make a meaningful impact to improve lives. We encourage you to respond to our flash surveys and other engagement opportunities to help us elevate the independent patient voice and change the status quo kidney care. You will also receive a digital subscription to our bi-monthly magazine, AAKP Renal Life, and can select to receive any of our 5 different electronic newsletters, all for just signing up. We also invite you to check out our blog to read some great articles and patient profile stories, and follow us on social media for all the latest news and to keep up on what's trending. AAKP is dedicated to helping kidney patients across the disease spectrum understand their condition and take control of their healthcare. We are proud to offer a variety of resources for both patients and care partners. By visiting our website at aakp.org and clicking on the AAKP Store button at the top of the homepage, you can find a variety of educational brochures and online tools to order or download free of charge, including titles such as the Understanding Kidney Transplantation brochure or the Kidney Transplantation webpage. You can also order materials by phone. AAKP has a number of exciting events planned for 2024, including our 6th annual Global Summit on Kidney Disease Innovations, hosted in partnership with the George Washington University School of Medicine and Health Sciences, which will take place virtually July 16th and 17th. Our 7th annual Policy Summit is scheduled to take place November 20th at the National Press Club in Washington, D.C., and will also allow for virtual participation. Registration for both events is now open. AAKP's 49th annual National Patient Meeting will be a hybrid using AAKP's sophisticated virtual event platform, with the in-person component taking place in September in the beautiful state of Florida. The dates are to be announced. And finally, you can also visit our YouTube channel to watch a number of great presentations and webinars, all available on demand from our 2023 events and programs, as well as 2024 programs as they occur. Once again, a big thank you to our webinar speakers, Dr. Mike Curtis and Dr. Shuvo Roy, for taking part in today's HealthLine event. The webinar was truly fascinating and shed light on the future of transplantation. We are grateful for everyone's contributions and for making this event both meaningful and informative. Thank you to all who joined us. Have a fantastic day and continue to celebrate National Donate Life Month with AAKP.